Thank you, Mr. Chairman. Thank you, Dr. Khanna and the organizers for inviting me this evening to this meeting. Now, I know that it has been a long day. I don't know whether you have got the patience to listen to me, but I'll try to be as provocative or say as exciting as possible. Now, the topic this evening I'm going to discuss is the anticoagulation treatment that has been developed over the last 7 decades, and without which we would not have been in this room to discuss about catheterization, vascular surgery, cardiac surgery, or any of those things. So let us show our gratefulness to the anticoagulation treatment. Let's have some patience to listen to my talk today. Now, in this talk, I'm going to strictly confine my discussion on venous thromboembolism. I could perhaps discuss on arterial, but there won't be much time to discuss this further. As we know that life has been saved, both due to venous thromboembolism as well as arterial thromboembolism, cardiac embolism, and also diseases associated with embolism such as cancer, pregnancy, or hemodialysis needs. Now, despite all these anticoagulation that we have been using over the last 7 decades, particularly heparin and warfarin, we are now looking for a need, the pursuit for a new anticoagulation treatment that can not only be effective but also safe. And to that end, what we are looking for in an anticoagulation agent is that it should be ideal. So what are the characteristics of an ideal anticoagulation agent? That it must be safe, it can be given orally, and it doesn't have any major interaction with the commonly used medications we use in our day-to-day practice. And finally, not finally, of course, it should not have severe adverse effect, and it should be available cheaply. Now, all of us know that heparin and warfarin have dominated the scene of anticoagulation treatment over the last 7 decades. And they are cheap, but they have got many side effects. And that led us to the research of finding out other agents which can be much more easily used, cheaper, or they can be safer, or they can be more effective. Our group in the 1980s and 90s, led by Professor Vijay Kekar, developed the compound called low molecular weight heparin, as well as the ultra low molecular weight heparin. And today, low molecular weight heparin is probably the largest used medicine in the world, particularly as an anticoagulant, as a thromboprophylaxis, as well as some treatment. Similarly, pentasaccharide, which is the active molecule of the heparin molecule, has been put into practice as fondaparinux, and which has been shown to be very effective in various as a thromboprophylactic agent. But what we are interested in talking about today, this evening, is the new oral anticoagulants. And those are either the group which belongs to the thrombin inhibitors or the group that belongs to the factor Xa inhibitors. So there are 3 compounds that are available as anti-factor Xa compounds, and they are rivaroxaban, apixaban, and idraparinux. However, on the thrombin inhibitor side, there is only one compound that is being developed today. After the failure of jymalagatran, which was developed in the 2005 and 6, the dabigatran has stood the test of the time, and the side effect that was associated with jymalagatran was obviated due to the development of dabigatran. So I will discuss these compounds mostly and in connection with the venous thromboembolism. Before we go to discuss any further about these compounds, let's understand a little bit about their pharmacokinetic and pharmacodynamics. And to begin with, let's see, where do they act? What is the mechanism of action? Heparin and warfarin, they have effects in the clotting cascade, particularly most of the factors that involved in the clotting cascade. Particularly on the heparin side, it is more effective over the thrombin as well as factor Xa and the other clotting factors. Similarly, on the warfarin side, their effect on the 2579, and as a result, they affect our global clotting assay. And therefore, when you do the APTT, particularly in the case of heparin, or the INR in case of warfarin, these results are delayed and they indicate the risk of bleeding. Whereas the selective agents such as factor Xa inhibitor or thrombin inhibitors, they specifically act on the factor Xa or the thrombin, and as a result, they do not affect the clotting casket or clotting assays, particularly the APTT or PT, as a result, and also, therefore, they do not cause any risk of bleeding. Now, here are the summary of the detailed pharmacokinetic and pharmacodynamic action. Dabigatran and apixaban, because they have got a half-life which is shorter than rivaroxaban, they are supposed to be given twice daily, whereas rivaroxaban has to be given once daily. Whereas the other interesting compound, which is idrabianoparinox, which has to be given only once weekly as a subcutaneous treatment, and this has been used for the treatment of DVT in one trial only. Furthermore, the other interesting aspect of this, that the currently available trials, particularly using dabigatran in venous thrombosis prophylaxis as well as treatment, has shown they're effective as well as safe in the treatment as well as prevention of venous thromboembolism. Similarly, the other compounds have also shown similar kind of action. Let us discuss those compounds individually. Dabigatran, as I said, it is an orally administered compound. The trials such as Remodel and Renovate have shown that they're equally effective as with enoxaparin in preventing venous thromboembolism in orthopedic surgery. And there was no significant difference with regard to side effects and particularly bleeding complications. Similarly, in the treatment arm, the Recover trial, the dabigatran again was found to be as effective as low molecular weight heparin and warfarin. And they were non-inferior to these compounds. And this has been now approved by NICE in UK for routine use in the National Health Service. Rivaroxaban, again, another orally administered compound, factor Xa inhibitor, and there also been trials like Record 3 and Record 4 trials in orthopedic surgery as a thrombosis prevention and venous thromboembolism prevention, and they have shown to be superior to enoxaparin, and they're also comparable safety profile. However, in this particular trial, the dose that was used in the use of rivaroxaban higher than other compounds that has been used in similar trials in venous thromboembolism. In Einstein trial, rivaroxaban was also shown that it is as effective as enoxaparin and warfarin in the treatment of acute venous thromboembolism. Again, rivaroxaban has been also licensed in UK for treatment in the NHS and prevention treatment. Similar results also have been obtained by apixaban in advanced trials, but however, here the efficacy or the effectiveness in preventing venous thromboembolism is much higher compared to enoxaparin. And they have also got a similar bleeding side effect profile. Idraparinox, as I said, this is a compound which can be given only once in a week, and therefore it is good when you are looking for long-term treatment of venous thromboembolism as well as in the prevention of thromboembolism in treatment of arterial fibrillation with warfarin. And there are some interesting results from this, but it has not been tried largely in clinical trials as much as the other compounds that I described. So these new anticoagulants, thrombin inhibitors and factor Xa inhibitors, the good thing, the advantage of them is that we need only fixed daily dosing because their effect is not altered by various other concomitant use of other medications. They can be given orally. There is a rapid onset of action, and they can be used in various situations, whether venous thromboembolism, arterial, or other, or following malignancy. And there is no need for monitoring the blood. But unfortunately, there are many things they have addressed in these trials. For example, there is no specific antidotes which can antagonize the effect of these compounds, particularly in an emergency situation when the patient needs to go to the theater. And the cost-benefit analysis is lacking, and these compounds at the moment which are available in the market are very expensive. And there are also limited data available for prolonged long-term effect of this use of these compounds. So now, in conclusion, that we have got factor Xa and thrombin, but the fact is that which is superior? We don't know unless we are doing some head-to-head trial. But the fact we know that one molecular factor Xa inhibitor can inhibit 1,000 molecules of development of thrombin. Therefore, it makes sense to try factor Xa rather than thrombin. I think that's all I need to say this evening. Thank you.